As we continue on here, we're going to start looking at some of the other organelles now. So we had kind of been working through the organelles to start with in the first half of this. So we will finish up some of those, talk a little bit about mitosis, and then, like I said, we'll move on to the actual different tissue types, and from there get into the organ systems. So we talked about the Golgi at the end here. The next one to kind of talk about is the mitochondria. This is another membranous organelle. It's not part of that endomembrane system where a lot of the other organelles can swap membranes with one another. This is its own organelle that kind of stays completely together. There's enzymes that are part of the Krebs cycle and the electron transport chain. There is a nucleus to this one that is, a chromosome to this that is slightly different in structure. It has its own DNA that we refer to as mitochondrial DNA, and it houses really the ability to generate ATP from a number of different nutrients in the cell, at least a large amount of ATP. Outside of that, we can talk about different surface modifications. Certain cells that we will look at, cilia, are present on the surface. These are long, thin projections that are going to, generally in the human body, move things past the cell. Flagella are usually larger. The only cell that we would look at that these are present on would be sperm cells. Cilia, usually more of them and smaller than a flagella. They are gonna, to me, I always kind of joke around and kind of say it's like crowd surfing. All the little hands are like the flagella if somebody's crowd surfing. Where flagella, it's gonna move a lot more like how an alligator or crocodile swims with that swaying tail side to side. They are made up again of these microtubules. They have this 9+2 arrangement, 9 around the outside, 2 in the center. Again, a lot of these movements are by using cellular motors to pull these microtubules in different directions past each other. Another surface modification we'll see are what we refer to as microvilli. These are not for movement. These are actually increasing the surface area of the cell. These are very tiny projections that there's probably thousands or hundreds of thousands on the surface of the cell. These are going to really increase that surface area. And again, anytime we're increasing surface area, it's going to increase the ability to bring things into that cell. Anything that requires any type of transport or a membrane to get across, if we have more membrane, you can have more transport proteins, and therefore it can do more absorption. A lot of times we see these in areas where a lot of absorption takes place, things like the intestines, things like the kidneys. There is some specialized types here called stereocilia. These are long microvilli. Place most common to see these is actually in some of the inner parts of the ear, where your vestibular senses and your hearing senses are present. Another thing we see a lot of in cells is these different cell junctions. So we have what are called occluding or tight junctions, anchoring or adhering junctions, as well as what we refer to as gap junctions, ones that allow signals to move from one cell to the next. These occluding and adhering junctions are ones that are going to make tight seals between either epithelial cells or attach these cells to one another, making these rings of attachment in terms of the anchoring junctions. Gap junctions, like I said, have a small pore in them. I see these, probably the most obvious places in cardiac muscle cells that are letting the signal from one cell move to other cells. The other thing we can see inside of cells, a lot of times there's going to be these occlusions. These are areas where we store a lot of molecules. A lot of times you'll see glycogen granules in here. These would be in a cellular inclusion. Fat cells, you'd see an adipose cell doing that. And you can see that on this one here, all these little secretory granules on the left arrow there. Each one of those little dots in there is that granule. And you can see if you do an electron micrograph, you can see them a lot larger. But again, sometimes these will stain and will be distinct inside of a cell, and that's why we point them out. Another thing that's kind of important to us is the cell cycle. You do have the different stages of the cell cycle taking place. We will probably not focus on this too much because you do this in other classes, but there is the different stages of mitosis. Interphase, which is a combination of that G1 phase, the kind of getting ready for the move, the S phase, which is the Xerox, the copying of all the DNA phase, and then G2 is that kind of synthesis of the stuff that is needed for the cell division. That gets us into those stages of mitosis where that copied genetic material is going to get moved into two separate cells, which are separated by cytokinesis, which is that cytoplasmic separation of the two daughter cells. When we actually look at mitosis, there's really four phases to that. You can kind of talk about an in-between one and give it a fifth phase. But during prophase, and I a lot of times then use the analogy of picking teams on the playground for this. Prophase is kind of figuring out who's playing, so the chromosomes condense, the captains get figured out in terms of those spindle apparatuses and the centromeres becoming prominent. We then are going to have everybody line up. That is metaphase. Prometaphase is kind of the disintegration and getting ready for that metaphase. So you can see prophase here, chromosomes are condensing. When we get to metaphase, all these chromosomes are going to line up at the center or equator of the cell. So that's what's happening during that metaphase. You can see it on the top one right here. To me, that's having everybody line up. We are then going to have the captains pick teams. So those two centrioles are going to start separating the sister chromatids to each end of the cell. That is anaphase, or the picking teams phase. You can see it in a micrograph right here. Those sister chromatids getting pulled to the two sides of the cell. I think I went the wrong direction. Yep. So then telophase is where the nuclear envelope is going to reform. A nucleolus reappears. The spindle apparatus and those centromeres are going to disappear. To me, this is kind of having everybody huddle up before you start the game. And then, like I said, cytokinesis is the finishing of this cleavage furrow to separate these two cells into daughter cells, which you can see is a little bit further along right here. So you can see that telophase separating the genetic material apart. That is those stages of mitosis. Again, this is not uniform between all cells. Certain cells are almost always continuously dividing. There is other cells that are hardly ever going to divide. Things like muscle cells and neurons really don't divide hardly at all. Certain tissues, you can see different stages of this taking place. A lot of times you won't see it as much. Main thing here, cells, a lot of different components to them. Some are more prominent than others in certain tissues, and that's why we talk about this now, is when we get to these certain areas where we're talking about a prominent rough endoplasmic reticulum, for example, in the Nissl bodies in the nerve cells, or something else in a different cell. That is why we kind of point out some of these organelles here. Main thing is, cells are complex. They have certain things they need to do. Certain ones are going to divide, other ones are not. But it's really the combination of how these cells work that are going to give us our tissues. And again, with these different cell types and different tissue types, is going to allow us to have these different organs with multiple cell and tissue types taking place in there that allow things like the stomach, the intestines to have their functions that they have. So this is kind of the end of the two intro chapters here that are really taking a look at kind of the basics of this field of histology, as well as the basic understanding of a cell. What we're going to do from here on out is we're going to start talking about the basic tissue types. So things like, we'll start with epithelial tissue and go through the different types of connective tissue, and finally finish up with blood and muscle. And that's what we're going to be working through for these first few sets or first few chapters before you're going to do your first test. So I will talk to you again about epithelial tissue.